Yeah, you'd probably like cellular neurobiology too.
Okay.
325. So, sure. I'd like to be done.
I was thinking that actually, as everyone was leaving, I was like, oh, no.
Nope, gotta keep going. Hi. Hi. How's your world today?
Good.
Good.
Yeah. I got to sleep in. It was nice.
Got an extra 20 minutes.
Didn't have to be here at the bus crocodile. Talk to me soon.
Oh, that's right. You were here at 7:30 yesterday. That's painful. And you didn't get out of here until 9:30 last night, or 9 last night. And then only went home to the cats.
It's okay. Called Josh on the way home. All is well. Oh, I told him that you were asking about like how you make the
towers?
He's sending pictures.
Okay.
Apparently they've done a test recently for not a cone but a cylinder. So they have the machine running that much. He also says that he has no idea in general about towers.
He just knows how to make this machine to weld them together. Do they have to make them conical? I guess they want to take up less volume and take up less material to build them.
Right.
And the strength has to be big at the bottom, like a big base, and taper to the top.
Yeah, I think it's probably more stable too, but I don't know.
Yeah, maybe more stable, right.
He does say that he doesn't think they're big enough to fit a elevator in the middle because there's all of the cords running through the middle to harvest.
Oh.
Which I didn't even think about.
Hi.
I'm Ella. I just wanted to introduce myself. I saw that you were researching for like neuroscience. I was wondering if you're doing that on campus.
I don't have anybody in my lab these days. My lab is pretty much defunct. All I do is teach.
Okay.
So I can't take any students on.
Just wondering. I think it's super interesting. I'm part of the Honors Undergraduate Research Scholars Program.
Okay.
So I'm always looking for more.
Talk to Katie. Katie Brown was ahead of that for a while.
I will. Thank you.
And give. She's often in here. She's not here today. But talk to her about that. She'll have real interest in it. She has interest in anything, but talk to her about it. She'll be able to give you some insights and places to go for doing some research of neurobiology, if that's what you're interested in.
Yeah, I am. I'm currently researching with Dr. Jessica Witt. She's doing, it's basically like how to communicate data the best way to people, which is super interesting, but I really like neurobiology, so.
All right.
Always keep my options open. But.
There's some possibilities.
Sorry?
You might also talk to Sassapa.
Okay.
Yes.
She's in here very often. She's hard to find, but she has office hours.
Okay.
And she's interested in microbiology. She's actually working in a microbiology lab where she's working on microglia.
Wow. Okay.
And trying to sort out one of the questions I brought up today, the difference between resident microglia and microglia that wander in from the periphery. Because the microglia are just macrophages, and she's beginning to find there are a gazillion different kinds of macrophages and many more different proteins that are affecting where they come from, where they go to, and where they settle down. And it is really interesting.
Okay.
And
I will talk to her as well.
And she's interested, and that lab is interested in, particularly in tuberculosis.
Really?
And there are invasive forms of tuberculosis into the central nervous system, and the only form of defense in the central nervous system for immunity are the macrophages, or the macrophages, or the microglia. And so the question is, how does infection change them? And how does it change the expression of various proteins?
Wow. Okay. I'll definitely.
So definitely track her down.
Yeah, that's cool. Thank you so much.
Oh, sure.
I'm really
Sure.
enjoying this class.
Oh, good.
I thought it would be way too much, but it's really cool. I
Good,
love it.
good.
Thank you.
Sure.
said, so it's 300, well, it would be, yeah, a second. What was the rate? So millimeters per day? Well, that's not even a foot, that's 3 to 4 centimeters.
Yeah, it's like 30 centimeters a day.
30 centimeters.
Right.
Okay. I was wondering, I was like, maybe my dimensional analysis is off.
And one is moving at 0.5 to 2 millimeters a day.
Yeah, so that one's like really, really slow because the difference between those numbers is enormous.
That's in the cell
The cytoplasm is, well, the slow transport is moving all the way down the axon at that slow rate. Yeah. And I'll finish that story today.
It seems just terribly inefficient though.
Well, think of what you have to use to move it.
Yeah, exactly.
You're using an individual molecule
Yeah,
that's taking 8 nanometer steps.
yeah.
So so divide it all up, and I've done these numbers before. I can't hold them in my head.
Yeah.
Figure out how many seconds there are in a day,
Yeah.
and then realize that in those seconds, during that period of time, we've taken 8 nanometer steps to get that distance.
Yeah, well, I mean, even if a 1 nanometer step is, millis, right? Divided over that is, it's moving at almost an evaporator speed.
I know.
Those little,
Yeah,
exactly.
I know.
But it just feels like, when you look at those numbers, it feels like there should be a selective pressure to make something.
To make things go faster, but I think you're limited by the rate of the enzyme to make ATP into ADP.
several days in advance.
Oh yeah, weeks in advance. Right, so the proteins that are now arriving at the tip of your toe were probably made, particularly slow exoplasmic transport, probably made, what, a month ago? I have to look at the numbers, but if it's a meter, it's 20 days ago anyway. Yeah. Oh, shoot. Okay, so we can get started here. So yesterday we closed with a discussion of the rabbit retina. So I'll bring that up today and try to emphasize why that's so important. But mostly what I want to do is I want to spend today's discussion on glia, even though we're going to spend a little time on slow exoplasmic transport. Most of the discussion will be on glial cells. And glia have gotten a bad rap for the last 100 years, more than 100 years, because we didn't really know how to measure what they do. We're just beginning now to begin with molecular biology techniques to be able to figure out the kinds of proteins that these glial cells are expressing and understand their function in the central nervous system. Up to now, most of our measurements have been on neurons. We can measure what neurons do really well because we have tools and electrical tools and amplifiers to measure the movement of ions across membranes. So the electrical tools have biased our understanding of the nervous system in terms of neurons. And we're just now beginning to see the depth of possibilities of what these glial cells can do. Turns out, and I'll mention this in the discussion, that there are probably 9 to 10 times as many glial cells as there are neurons. So there are way more glial cells than neurons, but yet we tend to put them in the background and emphasize the role of the neurons. Now, one of the things we'll see as we go through this discussion is that the astrocytes in the central nervous system particularly play a role supporting the neurons. They're going to play the role of controlling the extracellular environment. They're going to control the concentration of neurotransmitters at synapses. So they're regulating the environment in which the neurons function. And one way of people have thought of this is the astrocytes basically are the housekeepers. And the neurons are kind of prima donna cells that can't do much for themselves at all. And so they have to be maintained and looked after by the astrocytes. And that's probably to some extent true. But I think as time goes on, we're going to see long-term forms of communication from astrocytes to neurons and from astrocytes to other astrocytes that are playing an important role in the health and viability of the nervous system. The next character we'll talk about are oligodendrocytes. Again, these names are left over from the 19th century when the anatomists first looked at them. They thought the astrocytes looked like stars, so they called them astrocytes. And when they looked at the oligodendrocytes, they said, well, they just have a few branches on them. So they're oligo, meaning few, dendro, meaning trees or branches, cells. We know now they're pretty highly branched, but I'll tell you a story of what their function is. Their function is to provide an insulating layer around the conductal regions of neurons. And we'll spend a little bit of time talking about how these insulating layers are laid down and how they function to increase the rate at which neurons can signal one to the next. The microglia are different characters altogether. In fact, I'm always reluctant to include them as the glial cells because the microglia are simply macrophages. The microglia have originated from mesenchyme. They've originated as a kind of hematopoietic stem cell, whereas these two cell types, along with the neurons, come from ectoderm during development. So they have very different lineages. We're now recognizing that the microglia set up housekeeping in the central nervous system during development. There appear to be a set of microglia that are resident microglia, they never leave, and another set that will sometimes wander in and wander out. But for the most part, the microglia are set down and set into the central nervous system during early forms of development. The microglia are the only form of the immune system we see in the central nervous system. If things are working properly, the central nervous system is isolated from the immune system. It's said to be immunoprivileged. The central nervous system does not see the immune system. We'll see probably on Monday that when the immunoprivilege of the central nervous system is violated and the immune system can get into the central nervous system, one of the things that often gets attacked are the oligodendrocytes. And the destruction of the oligodendrocytes in the form of multiple sclerosis is devastating because it breaks down the oligodendrocytes and prevents them from insulating the axons and the movement of electrical signals. And we'll talk a little bit about that. But when we talk about glia, we think about glia in these two different realms. One realm is in the central nervous system. That's the astrocytes and the oligodendrocytes and the microglia. But the other realm is in the peripheral nervous system. And in the peripheral nervous system, there's a different cell type altogether. It's called a Schwann cell. It even comes from a different embryological origin. It originates in a region of the region of development called the neural crest. That's a detail, but these cells come from a different place than any of these or even other neurons in the central nervous system. Schwann cells are sometimes called neurolemmacytes. I think there's only one M in there. The neurolemmacyte is the same thing as the Schwann cell. Technically, I should use that term because it describes what it is. It goes around the lemma, the membrane of the neuron. But I just find it such a mouthful that I stick with Schwann cells. When I'm talking about Schwann cells, I mean neurolemmacytes. Again, the role of the astrocytes is basically housekeeping. And we'll see one of the first things it does is it regulates the components of the extracellular space. And until you've looked at thin sections or sections in the central nervous system, you can't really recognize the importance of how little extracellular space there is in the central nervous system. In the peripheral nervous system, there's lots of extracellular space, but once we've crammed all these neurons and glia inside our skulls and inside the spinal cord, there just isn't much extra space. And so we have very limited extracellular space in the central nervous system, and that's the role, that now becomes the role of the astrocytes to regulate and control the milieu of that extracellular environment. The oligodendrocytes have a different role. What they do is they lay down myelin. They form these insulating layers around axons. And as forming these insulating layers around axons, they improve or increase the conduction velocity of signals in neurons. These are always wrapping the conductal regions. They don't have anything to do with dendrites. They just wrap the conductal regions. Now, one of the things we'll see is that the oligodendrocyte is going to wrap round and round and round the axon, but it doesn't do this continuously along the axon. It only wraps a segment of the axon of maybe a millimeter and a half. And then there's a break. And in that break between oligodendrocytes, there's a region known as the node of Ranvier. This is a really important site. The node of Ranvier is basically naked membrane of the axon peeking out into the extracellular space. So we'll have rolls and rolls of myelin around the axon for about 1.5 millimeters, and then we have a break, and then we have open membrane of the axon, and then we begin again. And we'll see that multiple sclerosis now is a disease which causes the destruction of these oligodendrocytes. And finally, probably not today, but probably on Monday, we'll talk about Schwann cells in the peripheral nervous system and how these generate pretty much the same function in the peripheral nervous system that oligodendrocytes do in the central nervous system, and that they ensheathe the peripheral conductal regions. They have nodes of Ranvier, they have internodal regions. But one of the major differences between Schwann cells and oligodendrocytes is where oligodendrocytes in the central nervous system inhibit neuronal growth. Schwann cells in the peripheral nervous system will support it. This is clinically really important because we would really like to have a means when we break neurons in the central nervous system of getting them to regrow and go back to their normal sites of innervation. But whenever we break these neurons in the central nervous system and try to get them to regrow, they stop regrowing immediately, and that inhibition of growth largely is because of oligodendrocytes. These cells in the periphery, the Schwann cells, don't have that function. Instead, they're much more likely to support regrowth. And so what they'll do then is they'll permit and sort of nurse the growing axons back to their original sites. And there was a question up here. The oligodendrocytes make up the myelin sheath. We'll go through this in some detail. What the myelin sheath is going to be is it's an extension of the cytoplasm surrounded by the plasma membranes of the oligodendrocyte. And that extension of the cytoplasm with the membranes is now going to attach to the axon and go round and round and round. And every time it circumnavigates the axon, it leaves behind its two membranes with a tiny sliver of cytoplasm in between. So you're laying down phospholipid bilayer after phospholipid bilayer. And each one of those phospholipid bilayers is a pretty good insulator. So now we can provide insulation around the axon. So let me quickly, since I'm a little out of phase, let me remind you where we ended off last time and the importance of these experiments. Where we left off was to talk about the rabbit retina. And I'll remind you again that in the rabbit retina, there were a series of neurons we were interested in, although there are a whole bunch of different kinds of neurons in the rabbit retina, or in any retina. The ones we were interested in were these so-called retinal ganglion cells. These retinal ganglion cells obey all the rules we talked about yesterday of neurons. They have neuronal cell bodies out here, and they have axons, processes that they extend during development into this region known as the optic nerve. So if we were to superimpose the story from yesterday on this, and we used our pineapple neuron, we could imagine this is a pineapple input region of one of those ganglion cells, and the process then would enter the optic nerve. We know where all of these ganglion cells reside. They're in the retina. We know where all of the axon terminals, the output regions of these neurons reside. And this is in a region of the brain called the lateral geniculate. Those are anatomical terms, but what I want you to recognize here is that we have a site where the cell bodies are located. We have these long segments in the optic nerve where we have the axons, the conductal regions. And then out here, there's a point where those neurons terminate and form synapses onto other cells. So we have the full panoply of the structure we talked about yesterday. But not only do we have the structure, but we can also utilize the anatomy here of the retina, because it's near the outside world. So we can imagine taking a needle, poking the rabbit's eye with the needle and injecting amino acids. But these are radioactive amino acids. Those radioactive amino acids will be taken up by the cell bodies of the retinal ganglion cells. These radioactive amino acids in the retinal ganglion cells will then be converted into proteins. And then we can ask the question, what's the fate of those proteins once they're produced in the cell bodies? What's the fate of those proteins as they move along the axons? This is the first indication of how proteins were synthesized in the cell body, put onto transport systems, and moved down the axon. And the advantage of that is that by keeping good records, they could inject radioactive amino acids at a certain time, have those radioactive amino acids converted into proteins, and then wait for hours or days, kill the animal, dissect out the optic nerve, and then simply take sections, cut sections of the optic nerve, where these are sections, and ask the simple question, do we get radioactivity in these sections? And I think you can see that immediately after we inject radioactive amino acids, before they've had a chance to make proteins, there won't be any radioactive amino acids in those axons. But wait a few hours or a few days, those amino acids will be converted into radioactive proteins and moved down the axon in some sort of transport fashion. So what they could do then is they could dissect out the various optic nerves, knowing the time between when their radioactive amino acids were injected and where they were along the length of the axon, and ask the question, what kind of proteins appear? They could then map this as time versus radioactivity. And when they did that, what they discovered is they had 2 peaks of protein. And the longer they waited, these 2 protein peaks got farther and farther apart. And from that, they recognized that proteins were moving as a fast component, and they could establish that this fast component was moving at a rate of about 200 to 400 millimeters per day. Whereas the slow component was moving at a rate of about 0.5 to 2 millimeters per day. So rather than just giving you these numbers, this is how these numbers are established. They were established by this critical experiment of understanding how the radioactive amino acids were being moved as proteins in the axon. And they could see that they were moving at 2 different rates. They then further drilled down into that and found that the fast component were vesicular proteins. So from our discussion of cell biology, you'll know that these are transmembrane proteins, proteins destined for secretion, and perhaps even lysosomal proteins. And they're moving down the axon in vesicles at a fast rate, whereas the slow component was predominantly cytoplasmic proteins. And the cytoplasmic proteins, which included things like the tubulin and actin and the glycolytic enzymes, all of those were moving at the slow rate. Now, the history of this was sorted out, probably in the mid to late 70s. And the final story of kinesin and microtubules was sorted out in a few years. That was pretty much done in a couple, 3 years. But the question of what was driving the slow component took another couple of decades to sort out. At the time, it was thought that the slow component was simply being assembled in the cell body, such as the intermediate filaments and microtubules being assembled from tubulin. It was thought that these were assembled in one place and just pushed down the axon by the assembly of more protein, as though you were pushing toothpaste out of a toothpaste tube. Turns out that's not right. Turns out that's completely wrong. And what we find is that the slow component moves intermittently. When moving, it moves at fast rates. The mechanism of this is not entirely understood, but what we do know is that if we take proteins in the axon and we label them, so this is meant to be the axon membrane, and inside in blue, this is the cytoplasm. In ways that are a bit mysterious, it appears what happens is that there are enzymes that will cut out a piece of the cytoplasm. So I think of it as a kind of raft of cytoplasm. And that raft of cytoplasm then will be attached to green. Will be attached to a microtubule. And that bit of excised cytoplasm will be attached to a kinesin. And the kinesin now will propel this little chunk of cytoplasm along the microtubule. Again, as a plus-end directed motor. And while it's attached to the microtubule, this fragment of cytoplasm will move at fast rates. It will move at 200 millimeters to 400 millimeters a day. So, well then, how do you explain this disparity between the fast and slow rates? And the answer to that is simply that when these proteins are moving along the axon, they're moving at fast rates. But they only move along the axon intermittently. So they get on the axon, travel for a while, get off, get intercalated back into the cytoplasm in ways we don't understand, and then they stay put for a while. Then a piece will be cut out, it'll get on the microtubule and be transported at fast rates. So they're moving. When they're moving fast. But most of their time is spent standing still. Several years ago, there was a GTA in this class. And I think she came up with a great analogy of this to think about it. And she said, well, this is a little bit like trying to hitchhike to Albuquerque from here. So you can imagine you go out to I-25 and you stick up your thumb and you wait for a bit and a car stops and picks you up, but the car says, I'm only going to Berthoud. So you get in the car and while you're moving at the same rate as the rest of traffic. You're moving at 70, 75 miles an hour. So you're moving at fast rates. But you get to Berthoud, you get out of the car and you stick up your thumb and you wait for another ride and you're standing there all that time. You get back into the car. When you get in the car, you're moving at fast rates. But maybe you only go as far as Longmont or Denver. And you get off and you wait again. It's a good analogy for what's going on for these bits of cytoplasm. They're being cut out, and when they're moving with the rest of the traffic. They're moving the fast traffic. They get off, and they stand by the side of the road until they can be cut out and picked up again. And that's the difference between these fast and slow rates. And that puzzle took a long time to figure out. So any questions up to there? Yes?
Do we know what makes them start moving again? Why they start moving?
Why they start moving, I don't know that it's completely known. I think it's probably a series of events. My simplistic view of it is that you have a chunk of cytoplasm, it gets attached to a kinesin molecule, and off it goes. It's basically like loading it on a truck, and it's going to move with the traffic until it gets unloaded. I think it's just a question of being attached and off it goes. It can't help itself. Yes?
So I'm kind of confused about what you mean by like a chunk of cytoplasm. Because I thought cytoplasm was like fluid and ubiquitous. What exactly?
So Fair enough. I mean, what do I mean by cytoplasm? Well, if you'd asked me that question, maybe 50 years ago, 40 years ago, I would have said it's like chicken soup. You have these proteins floating around in this broth, and there's no structure to it. But as we began to understand from the ultrastructure of cytoplasm, we find that it's not just chicken soup. It's a highly structured series of proteins. And then within those proteins, there are soluble proteins, sure enough. And there's a lot of water. But even the soluble proteins are probably in some way associated by charge-charge interactions with other parts of the cytoskeleton. So it isn't just fluid, which is easily sloshed about. It's a pretty rigid kind of gel. It's really, rather than thinking of it as chicken soup, it's much more like Jell-O. And within that Jell-O are all these cytoskeletal proteins. So you can imagine taking a piece of Jell-O, cutting it out, putting it on a microtubule, and moving it for a while, and then putting it back into the Jell-O. That's the way I think it's best thought of. Yeah?
So for the fast components, are they just moving all the time?
The fast ones are, yes. Let me do the simple answer. Yes, they're moving all the time. But I think you're recognizing that if we were to lay out a whole series of these microtubules, they have to come off of one microtubule to get onto the other. So there are times which they'll be off the microtubule, but probably not very long. And then they'll reattach to a neighboring microtubule, and they'll start their movement again. Anything else? Yeah.
So would it be kind of like a handoff, from the different microtubules? Any center, like stationed at that certain microtubule?
So the question is it kind of like a handoff? And yeah, I think you could envision it like the handoff of a baton in a relay race. But it's probably not quite as deliberate in the sense that I suspect if you have another microtubule down here, and let's say we're actually using vesicles, so let's put proper vesicle on here. And it's moving along, it jumps off the end, and it's wandering around here for a little bit, finds a kinesin, and things are pretty close, so it probably doesn't diffuse very far, and will glom on to the next microtubule, and off it'll go. And as long as you have only kinesin available, it's only going to go in one direction. Anything else? Then let's move on to glia. So when we talk about glia, I'm going to use some generalities here, and talk about glia, and then think about glia and where they are. When we talk about glia, or glial cells, we find that there are something like 9 to 10 times as many glial cells as neurons. And to give you some sense of the disrespect that the early anatomists had for glial cells, the term glia comes out of the Latin meaning glue. They thought all they did was hold the thing together. They weren't really particularly important. We know now they have really important functions. But when we talk about glia as being separate and distinct in different places. We have one form of glia in the peripheral nervous system. And the glia in the peripheral nervous system have a name. They're called Schwann cells. Again, sometimes you'll see this referred to as neurolemmacytes, but most commonly they're referred to as Schwann cells. Then in the central nervous system, there's more variability in the types of glial cells. In the central nervous system, there are astrocytes and oligodendrocytes and microglia. Again, this term microglia was based upon their morphology. When the early anatomists looked at them, they said they were small, so they called them micro. They were in the central nervous system, so they called them glia. But in fact, the microglia are a cell type that's different, very different from the astrocytes and the oligodendrocytes. These are macrophages. You remember that macrophages are part of the blood system. So they originated initially from hematopoietic stem cells. And if you recall this discussion of connective tissue, this is going to be a type of connective tissue whose origin is mesenchyme. All that to say that these just are different cell types than the oligodendrocytes and the astrocytes. The microglia are the only part of the immune system that's functional inside the brain and spinal cord. The immune system cannot get into the central nervous system, as far as we can tell, because of structures known as the tight junctions of the blood-brain barrier. But the microglia, it appears, until recently, we thought the microglia could come and go. They were just macrophages that could come in and leave. But it looks like now the microglia are arranged in such a way that there are what they call resident microglia. They're set up at the time of development. So early on, as you're developing in utero, you'll set down a series of macrophages, and they'll live the rest of their lives inside the brain and spinal cord. Those are resident microglia. There are others which seem to come and go, but they seem to be less important in the immune system. We don't quite understand them yet. But the microglia I'm going to leave behind because they're part of the immune system and emphasize these two other characters. When we think about the astrocytes, what I want you to bring to mind immediately is that the astrocytes are regulating the extracellular environment. The function of the astrocyte is to make sure neurons have a good environment in which to signal electrically. About all neurons can do is send electrical signals. That's what they're really highly specialized to do. And they can't take care of themselves very well. So the astrocytes are thought to play the really important role in maintaining the environment in which neurons can function. So to try to emphasize that, I'm going to make a drawing similar to what we had yesterday, where this box is meant to represent the output region. Of a neuron. And the black box over here is meant to represent the input region. And the space in between is the synaptic cleft. It's basically a gap between the presynaptic cell and the postsynaptic cell. Now, the function, as we alluded to yesterday, is to have vesicles fuse to the plasma membrane of the presynaptic or output region and release neurotransmitter into this very narrow gap. This very narrow gap has a very, very small volume, so it doesn't take very many molecules before you increase the concentration of neurotransmitter in this narrow space. And I think you can see that if on the postsynaptic side, in the input region here, if we now have these molecules we described yesterday as ligand-gated, ion channels. You can see if we were simply spilling out neurotransmitter here, that we would be opening these ligand-gated channels. There'd be no way to get them to close. They'd be open all the time. So we don't want that to happen. We want to basically regulate the concentration of neurotransmitter here in the synaptic cleft. So if you look carefully at synapses in the central nervous system, right next door will be a process, an extension of an astrocyte. So someplace far away, there's going to be the cell body of the astrocyte sent out a process. And my drawing doesn't do it justice, but this astrocyte process is going to wrap around the whole synapse. So if you think of this output region, input region as cylinders, and the two cylinders are coming close to one another, then the astrocyte is going to wrap around that cylinder and be right next to the synaptic space. That allows for transmembrane proteins in the astrocyte here, which will be neurotransmitter transporters. So the neurotransmitter that was released from the output region has opened the ligand-gated channel and allowed ions to flow in. We can now regulate the number of molecules of neurotransmitter in this region by having these transporters. So these will be neurotransmitter transporters that will take the neurotransmitter and move it back into the glial cell, back into the astrocyte. And in this way, we can regulate the concentration of neurotransmitter in these very narrow gaps. That's the role of the astrocyte. Make sure we don't get a buildup of neurotransmitter in these synaptic spaces. Now, as a detail, the most common of the neurotransmitters in the central nervous system is glutamate. The ordinary garden variety amino acid glutamate. The glutamate will be released from the axon terminal, bind to glutamate receptors, open channels, allow ions to move. What the astrocyte does is it binds the glutamate, brings it into the cytoplasm, and in the cytoplasm, the astrocyte converts glutamate into glutamine. For the biochemists among you, it's simply an enzyme that adds an amine group. But the importance of that is that glutamine cannot open ligand-gated channels. So what the astrocyte does is it releases glutamine into the extracellular space. That glutamine can't open channels, but the neuron can take up the glutamine and convert it back into glutamate. So that's basically the cycle that's under the control of astrocytes in the central nervous system. It's one of the ways it inactivates the most important of the neurotransmitters, glutamate. But something similar happens with almost all the other neurotransmitters. And one of the examples that you may be familiar with is serotonin. There are a series of drugs that we take, anti-anxiety and mood enhancers, which are serotonin reuptake inhibitors. And what they do is they inhibit these proteins that are normally moving the serotonin out of the synaptic cleft and into the astrocyte. And in that case, we think that in many people, there's a deficiency in the amount of serotonin in the extracellular space, and that leads to mood disorders. So we give people these serotonin uptake inhibitors, which allow the serotonin concentrations to remain higher in the synaptic cleft for longer periods of time. Yes?
So the astrocytes, they basically form reuptake, right?
The role of the astrocyte is to do reuptake.
Okay, and so they're releasing the glutamate as glutamine so that it's not taken up by the next neuron?
So see if I can reword that a little bit. The glutamate, which can open channels, You don't want to let that run wild. So the astrocytes play the role of basically, you can think of it as vacuuming it. It's kind of an exaggeration, but it's binding to the receptor, moving it into the astrocyte. But it doesn't help if the astrocyte simply releases more glutamate out into the outside world, because that would raise the concentration of glutamate in those small volumes of the neurons. So instead of releasing glutamate, it takes glutamate or converts it into glutamine, which can't open channels.
The neuron is able to take it in when it's necessary and then convert it back into glutamate.
Yes, exactly. So the neuron then will take up the glutamine. It has enzymes to clip off that amine group and convert the glutamine into glutamate. And so then you can start the cycle all over again. Sure. Anything else? Yes.
How is the glutamine absorbed by the neuron?
So the question is, how is the glutamine absorbed by the neuron? There's going to be another transporter here. Almost all these amino acids are going to move across the membrane by something known as sodium-driven cotransporters. We're going to run into those again when we talk about the kidney and talk about digestion, but they're also really important to the nervous system. So there'll be transmembrane proteins here on these neurons, which will bind a molecule of glutamine on the outside and a molecule and an ion of sodium. And when those two are bound to the protein, it turns inside out and delivers the glutamine to the inside of the cell along with sodium. It's called a sodium-driven cotransporter. Yes, and then I'll come to you. You first.
So the glutamate, the reason we don't want it to run wild is because it can open up those channels?
Basically, yes.
Okay.
There's a little more detail to that, but I don't want to make it more complicated.
Okay.
Yes.
So is glutamate like a protein, or is it another neurotransmitter?
No, glutamate is a neurotransmitter, but it's not a protein. It's true there are some neurotransmitters in the central nervous system that are peptides, but they all work through G protein coupled receptors. That's another story. This is simply the amino acid glutamate, a single molecule of glutamate. And that glutamate can bind to receptors and open the receptors to allow ions to move. So we'll run into a whole flock of different neurotransmitters. In the peripheral nervous system, the most common one is acetylcholine. The central nervous system, the most common was glutamate. But there's also dopamine and norepinephrine and epinephrine, particularly in the central nervous system, and also in the autonomic nervous system. So we have a whole raft of different neurotransmitters binding to a whole bunch more receptors. And those will then lead to effects. Now, let's not get too deeply into that right now. The acetylcholine and the glutamate both operate these ligand-gated channels. The others I mentioned work mostly through G protein coupled receptors, but we'll come to that. Sure. Yes.
I was just wondering what the word at the top of the drawing was, synaptic cleft.
Synaptic cleft.
Thank you.
Cleft is just the space between the neurons. Sometimes it's known as a synaptic gap. Okay. The other thing that astrocytes do is that they regulate, particularly the concentration. I'll make it general, concentration of ions in the extracellular space. Now, the two major ions in the extracellular space are going to be sodium and potassium. The concentration of sodium in the extracellular space is about 130 to 140 millimolar. The concentration of potassium in the extracellular space is about 4 to 5 millimolar. And we'll see next week that if these concentrations of 4 to 5 millimolar exceed this range very far, particularly if the concentrations increase to 5, 6, 8, 10, then this is going to have really bad effects on the neurons and their ability to generate and propagate electrical signals. So we really need to keep the extracellular concentration of potassium in this very narrow range. And one of the major roles of the astrocytes is to pump potassium into astrocytes. And we think in some cases to the blood vascular compartment. Now, what I want you to think about is that around all of these neurons, if we increase the potassium concentration, we dysregulate the system. It breaks. So anytime the potassium concentration begins to get much above 4 or 5, the astrocytes become very busy in taking potassium out of the extracellular space and sequestering it into the cytoplasm of the astrocyte. So the astrocyte plays this really important role not only in regulating the concentration of neurotransmitters at the synapses, but also regulating the concentration of potassium in the extracellular space, making sure it doesn't get very high and disrupt electrical signaling. So again, the role of the astrocyte in its simplest forms is to regulate the environment in which neurons function. I'm sure, given 5 or 10 or 15 years, we're going to find a whole bunch more things that these astrocytes are doing. And I suspect as time goes on, we're going to see that astrocytes are playing a really important role in a variety of diseases. We just don't know what they are yet. But again, as we learn more of how they function, we'll get a better idea and a better handle on how that works. Well, let me finish, and I'll finish in the back board, with the oligodendrocyte. The oligodendrocyte as a myelinating cell. When we talk about myelin, what we mean is the myelin is an insulating wrap around conductal regions. The insulating wrap around conductal regions in the central nervous system is laid down by the oligodendrocytes. Be laid down by Schwann cells in the periphery. But I want to spend a couple minutes here showing you how this happens in development. And there are a whole raft of controlling mechanisms. But if we simply look at how the myelin's laid down, we start out with this structure that I'll simply label as the axon or conductal region. And then off to the side, I'm going to draw another cell, and I'm going to call this an oligodendrocyte. There's its neural cell, there's its nucleus. This is an oligodendrocyte. Now in development, this oligodendrocyte will send out a process. It's a process of cytoplasm, but it will be bounded by the plasma membrane. So this is the oligodendrocyte plasma membrane. Now, there are many, many proteins, transmembrane proteins, in the membrane of the oligodendrocyte, and many, many proteins, transmembrane proteins, in the axon. And some of those are sticky to one another. So what we can do is get this tongue of cytoplasm surrounded by the plasma membranes to stick to the axonal membrane. But it just doesn't stick. It sticks, it finds itself stuck to the axon membrane and then continues growing. So if we look at the next step here, you can imagine if this is a time lapse, that this oligodendrocyte has continued to have its process grow around the axon. It's stuck to the axon, and it grows around the axon. Now what's going to happen is when it meets its original membrane here, it's going to insinuate itself underneath it, and it's going to circumnavigate the axon again. So it goes round and round and round. And each time it meets its original site here, it grows under it. What this yields is kind of an arrangement a little bit like this. So bear with me because I often screw up this drawing. So this is the last bit. This is the growing tip, as it were. attached to the plasma membrane of the axon. But there are plasma membranes of the oligodendrocyte over and over again, because this oligodendrocyte has climbed, has crawled around the axon many, many times. This is where I usually get into trouble. So if I try to follow the cytoplasm here, because remember, these are the plasma membranes that are bounding the cytoplasm. If this cytoplasm, we could imagine swimming through the cytoplasm here, and work our way around and round and round. And we come back here and we find that this goes back to the cytoplasm of the oligodendrocyte. What this tells us is that the oligodendrocyte has grown round and round the axon, and each time it circumnavigates the axon, it leaves behind 2 layers of plasma membrane. Each one of those layers of plasma membrane is predominantly phospholipids. Phospholipids are great insulators. So what you're doing is you're wrapping this axon with insulation, much like you'd take electrical tape and wrap a wire. But in this case, each time we circumnavigate the axon, we leave 2 plasma membranes behind. In that case, we can make many, many layers. And if you look at axons in the central nervous system, sometimes there are hundreds of these layers, providing an insulation around the axon. What we'll do next time is we'll go from this cross-sectional view, turn it at right angles, and see how these oligodendrocytes are arranged on the length of the axon.